In a DNA extraction, cell must be lysed to expose the DNA and the membrane lipid should be removed through the use of an appropriate detergent before precipitating the DNA with alcohol.
And RNA can be removed with protease and RNase respectively. Phenylchloroform extractions are widely used in DNA purification methods using a mixture of phenyl, chloroform, isoamyl alcohol on proportion 25:24:1. After centrifugation, proteins, lipids, carbohydrates, and cell debris are removed through extraction of the aqueous phase with the organic mixture of phenyl and chloroform. The upper aqueous phase contains DNA and it is collected so that DNA can be precipitated with ethanol and a high concentration of salts. RNA is an unstable molecule with a very short half-life once extracted and should be isolated prior to use. Lysis buffers are used to disrupt the cells and strong denaturants are added in RNA isolation to inhibit endogenous RNase. A common method for RNA extraction uses a guanidium thiocyanate phenyl solution to denature proteins and maintain RNA integrity. Phenyl can be adjusted at an acid pH to favor the permanence of RNA in the aqueous phase. Trisol reagent and chloroform can be used to separate RNA and DNA in different phases. In this method, isopropanol is used to precipitate RNA as opposed to ethanol.
Cell is the use of different reagents for each of the steps of purification, mainly the cell lysis because the cell wall and cell membrane have different components.
Cells have cell membranes that are composed of phospholipids, and to degrade them it is necessary the use of detergents or compounds that specialize in this type of molecular degradation. To degrade bacterial cells walls, it is necessary the use of enzymes due to peptidoglycan wall that cannot be degraded with detergents. The use of enzymes is also necessary to degrade plant cell walls because they possess a primary and secondary wall which develops in a propagated form to the cellulose microfibrils arranged in an orderly manner with a denser structure than the primary wall. There are some column-based nucleic acid purification and they work under 4 conditions. 1. Lyse. Cells are broken open. 2. Bind. Adding a buffer solution to the sample so nucleic acids can bind to the silica gel membrane in the column. 3. Wash. The flow-through is removed. 4. Elute. The elution with buffer removes the nucleic acid from the membrane and the nucleic acid is collected from the bottom of the column.
After isolation and purification of nucleic acids, it is necessary to quantify their concentration in the sample. Among the quantification methods, we can find the following.
Use of the UV spectrophotometry at 260 nanometers to determine the concentration of DNA in the sample. Another measurement will be needed at 280 nanometers to get the A260/A280 radio, which determines protein contamination in the sample. Use the fluorophores that become fluorescent when binding to DNA or RNA, and the intensity of the fluorescence will depend on the size of the target molecule. Running in agarose gel from samples in which it is known that the DNA concentration will be very low. This method will measure bromide-ethidium fluorescence in UV light and comparing fluorescence intensities to estimate the DNA concentration. Use the slow-to-blood method that uses specific probes which will be directed to target sequences in a nucleic acid sample.
Human genomic DNA and bacterial RNA were extracted using 2 different methods. Each sample was analyzed in a spectrophotometer at an absorbance of 260 and 280 nanometers. The absorbance and concentration results are shown in Table 2.
Genomic DNA results, based on the spectrophotometer results, indicate that a sample is contaminated with proteins since the absorbance ratio is lower than the accepted range, which lies between 1.7 and 2.0. On the other hand, the RNA sample ratio is way too distant from the recommended acceptable range, which is 2. 2 different kinds of nucleic acids were obtained from a human and a bacterial sample. To purify the DNA, the cell lysis was carried out with sodium acetate, SDS, and proteinase K, while the lysis S-test buffer and trisol were used to disrupt the bacterial cells. The main difference in the methodologies were the reagents used to eliminate the proteins. To extract DNA, a phenylchloroform isoamyl alcohol solution was used in order to solubilize DNA and RNA in the aqueous phase and separate the proteins in the organic phase. In this case, both nucleic acids were present in the upper phase. With the addition of chloroform, RNA, DNA, and proteins are separated in different phases. Finally, the DNA was precipitated with a solution of sodium acetate and ethanol, while the RNA was precipitated with isopropanol. Taking into account the spectrophotometric results, we can state that both samples cannot be considered as highly pure since both present contamination. However, the purity of the genomic DNA is significantly higher than the RNAs since the absorbance ratio is closer to the acceptable range. Also, we can affirm that there was contamination with protein because in the results of the electrophoresis it can be observed the contamination in the wells.
When observing the gel, the first line is 1 kb marker. The next two lines are the RNA and DNA samples respectively.
The DNA run has a dark black section which indicates RNA contamination, and the top line of this run is the DNA sample. The RNA is not as clear, yet it has the 3 RNA lines observed along the gel. The results obtained in the gel were not of the best quality due to several factors. When reading the results in the gel, there is contamination present in the wells which indicate proteins that were not fully degraded. When doing PCR with contaminated samples, there are many problems. Using low purity DNA as a sample for isolation of an interest sequence could lead to an unsuccessful PCR. It is possible that primers will not hybridize in the correct places or sequence. Contamination with RNA present high background noise during PCR and can hinder the DNA quantification. Salt contamination will affect the processivity of polymerase, which inhibits sequencing.
It has to be taken into account the nature of the sample, if it is eukaryotic or prokaryotic, since each type of cell requires a distinct protocol and therefore different reagents. From the diluted sample, the value of absorbance will be measured different wavelengths against a blank sample, and then the concentration and total quantity of nucleic acids can be obtained.